Hi students, yesterday we were talking about that is a mutation. That is part 1. What is mutation and other things. Today we have to talk about the major types of mutations, the sudden change. There are some criteria have been used and which are considered as a basis for classification of mutations. So the mutations may be classified based on the origin, or based on the cells in which they are taking place, or based on the nature of the function, whether the function has been increased or decreased or actually eliminated accordingly, and also based on the molecular change. What we studied already about what we have the nucleotide substitution, just what we have transition, transversion, deletion, etc. And also those mutations which affect the translation process. These are the 5 bases. Based on what we have origin, based on the cell type in which it occurs, based on the effects of function, based on what we have the molecular change in the DNA molecule, particularly the nucleotide chain, and finally those mutations which affect the translation process. What we have translation process that is also affected by the mutation. I will take the first 3 first and then we will go back to 4 and 5. Now based on the origin, we can classify the mutations into 2 types. One spontaneous, another one induced. Spontaneous means the one which occurs naturally. That means a mutation that occurs in the absence of known mutagen, without any mutagenic agent. If mutation occurs in nature, then it's called spontaneous. Now the second one, induced. In the case of induced mutation, the mutation is induced by what is called a mutagen, maybe a physical mutagen or a chemical mutagen which induces the mutation. So those mutations which occur in the presence of known mutagen, they are called induced. So anyway, based on origin, we have spontaneous, induced. This is a natural one, and this one we induce artificial. Now the second type based on the cell in which the mutations occur. Some mutations occur in somatic cells. They are called somatic mutations, or actually we can say they are occurring in non-reproductive cells, the body cells. If mutations occur in the body cells or non-reproductive cells, you see that one occurs in non-reproductive cells, then it's called somatic mutation. On the other hand, if the mutations occur in the germ cells or in the reproductive cells, say an example of sperm and egg, then it's called germ-line mutations. So based on the cell type, we have 2 types: somatic mutations that occur in somatic cells or body cells, and that is nothing but the non-reproductive cell, and germ-line mutation, the mutation that occurs in the reproductive cells, the germinal cells, other than the somatic cells. So this is another category. Now on the basis of what we have the effect on function, some mutations affect the function of the what is called particular actually activity, and based on the effect on function, we can have 4 different types of what we have mutations. Number 1, loss of function. In some cases, mutation causes the loss of function. The meaning for that one, it eliminates the normal function. It eliminates the normal function. So the normal function is eliminated. It's not being taken place because a particular gene is being knocked out, or null. Null means without. Knockout means a particular gene's activity is being suppressed. That is called knockout. It's being eliminated in doing the activity. So loss of function, and that is called knockout or null, and that one eliminates the normal function. Now the next one, hypomorphic. Hypo means below, hypomorphic. Now this one reduces a normal function. This one reduces a normal function. The next one, hypermorphic. This is opposite to hypomorphic. Hypo means low, hyper means above. So it increases the normal functions, reduces the normal function. The first one eliminates the normal function. The second one reduces the normal function. The third one increases the normal function. The activity of function has been increased. Now the last one, gain of function. A function is gained, but that function is not being actually that is used by the organism, because what is happening, expressed, such mutations are expressed not at a proper time. It is being expressed at incorrect time or not in a specific cell, in appropriate cells. So those mutations which normally actually being gained, which results in the gain of function, they are called ectopic expression. Ectopic expression means other than what we have the specific cell, they are doing their activity in cells what we are not expecting. So the mutation which is being expressed at what we have incorrect time, not at a particular specific time what we need, but it is being expressed at an incorrect time, and also in inappropriate cells. Suppose we want to have this mutation beneficial in somatic cells or in the epithelial cells, but it is taking place, for example, in connective tissue cells. So those mutations where the function is gained but the function is not being utilized by the organism, this is because the mutation expressed at an inappropriate, what is called time, that is the incorrect time, or we have inappropriate cells. That's about the three different types of mutations. We have to go for two more mutations, that is with reference to the molecular change, nothing with the change in the nucleotide. That is what we have, the nucleic acid. Another one, effects on translation. Some mutations normally affect the translation process, leading to the formation of abnormal proteins with the non-functional status, and such are called effects on translation process. Those mutations which affect the translation process. I will take that one. Now the fourth type of mutation is based on the molecular change, or on the basis of molecular change. That is nothing but change within the DNA molecule. So that is normally a nucleotide substitution. That change is nothing but nucleotide substitution. Now the first type transition, I already explained that one. Here a purine is replaced by another purine, or a pyrimidine is replaced by another pyrimidine. So a base pair in DNA is replaced with a different base pair. The different base pair is nothing but actually another purine only, not a pyrimidine. You see that one? A purine base is replaced by another purine. For example, if you are taking adenine A, it is being replaced by G. And then pyrimidine to pyrimidine. That is, a pyrimidine base is replaced by another pyrimidine base. For example, T is replaced by C. That's about what we call transition. Now the next type, transversion. Here a purine base is replaced by pyrimidine, or a pyrimidine base is replaced by purine. Now you see that one, here purine to pyrimidine. I am taking A, that is a purine, the adenine, which has been replaced by thymine, a pyrimidine. Or a pyrimidine is replaced by a purine. You see that one, this is a pyrimidine cytosine replaced by what's called guanine. That's about transversion. Now this is insertion, that is a common one, addition. This is not the frame shift insertion or deletion, this is a common insertion of nucleotides. So one or more extra nucleotides are present, added, extra nucleotides are added, and that is called insertion, addition of one or more extra nucleotides. So we have one or more extra nucleotides are present. The next one, just opposite, deletion. One or more nucleotides are absent. One or more nucleotides are absent or missing, and that is what is called deletion, removal. This is addition, this is removal. So present extra nucleotides here, and extra nucleotides are not there, but some nucleotides are missing or lost. That's about what we have molecular change, what I described already. That's why I want to conclude quickly. So nucleotide substitution, we have transition, transversion, insertion, and then deletion, mutation. We have also insertion, frame shift insertion, that is different. And similarly frame shift deletion. This is a common addition and common deletion. Okay. We have to go for the last one, effect on translation process. The effect on translation process, I mentioned about the different types. You know, there is what is called a silent mutation, a nonsense mutation. We have what is called a frame shift mutation, addition and deletion. I already described, I want to just represent here the next one. Now the last type of major mutation on the basis of effect on translation. Some mutations affect the translation process, and based on this one we can have the following types. Number one, silent mutations, also called synonymous. So here I mentioned about, you see that one, a change in codon. A codon for an amino acid has been changed into another codon. That codon also normally coding for the same amino acid. There is no change in the structure of the protein because there is no change in the phenotype. This is because of the appearance of the same amino acid. That is called silent, synonymous. No change in amino acid encoded. Then missense. I mentioned already, you see that one. A codon, it is a mutation where a codon has been changed into another codon for a different amino acid. That is called missense or non-synonymous. Change in amino acid encoded. So we already described all these things, only the short form I mentioned here. Now the third type, nonsense. So when a mutation resulted in the formation of any one of the nonsense codons, UAA, UAG, UGA, and that is called nonsense mutation, also called termination mutation. So there are three codons you have studied which terminate the process of transcription, UAA, UAG, UGA. So we have here UAA, UAG, UGA. So when a mutation results in the formation of any one of these three codons, then we can say nonsense mutation or termination mutation. Then the last one, frame shift. So a change in the reading frame because of the shifting of what we have the nucleotide. So that what is happening, we have non-functional incomplete protein is being formed. So here it shifts triplet reading of codons. That is what we have, the open reading frame. So it shifts triplet reading of codons out of correct phase. So they are not present in the same phase because of what we have, they have been shifted either towards the left side or towards the right side according to deletion, or we have what is called insertion. So we have frame shift deletion mutation, we have frame shift insertion mutation. So in both the cases we have normally shifting of the nucleotides so that we have incomplete non-functional protein is being formed. So these are all the major types of mutations. Now we have to go for the next one, a simple thing about the mutagens. What are mutagens? What are the types of mutagens? Then what do you mean by co-mutagens? Like that, just I have to go through. Now the next one, what are mutagens or mutagenic agents? Now those factors that cause genetic mutation are called mutagens or mutagenic agents. And it was H.G. Muller, he was the first one in 1927 to find out mutagen in Drosophila. That means he induced mutations in the case of fruit fly by using X-ray, one of the physical agents, that is nothing but radiation. Now if you are taking the types of mutagen, there are two types of mutagens. One physical mutagens, another one chemical mutagens. Again the physical mutagens include number one, temperature, number two, radiations. Now what is the role of temperature? When the temperature is increased, the rate of mutation also increases. Increase in temperature increases the rate of mutation. Again you see that one, how it normally causes mutation. Rise in temperature causes the breakdown of hydrogen bonds between the two strands of DNA nucleotides. Two strands of DNA nucleotides or two DNA nucleotides. You see that one, we have the hydrogen bonds between A and T like this. This is the hydrogen bond. Now this temperature normally causes the breakdown of hydrogen bonds between the two DNA nucleotides. Between here A and T or G and C. That's a role of what is called temperature. Increases in temperature increases the rate of mutation. How it works? Now it causes the breakdown of hydrogen bonds between the two DNA nucleotides. As a result, what will happen? The transcription process and the replication process is affected. The transcription and replication process is affected. Both are affected. Now about the radiations, we have a number of radiations. The main one, for example, X-rays. X-rays, alpha rays, beta rays, gamma rays, cosmic rays. Even if we have the radioactive isotopes, UV rays, neutrons. These are all some of the radiations responsible for causing mutations. Now let's go for radiations. Now we are getting radiations from the electromagnetic spectrum. So they are nothing but electromagnetic radiations. These electromagnetic radiations contain short and the longer wavelength rays than what we call the visible spectrum. Now what are the types of, you see that one, electromagnetic radiations, and what are their effects, we'll see now. Now the second physical mutagen is radiation. So the radiation is in the form of electromagnetic radiations or waves, which have both short and longer wavelength rays, than what we have the normal visible spectrum. And these radiations are divided into number one, ionizing radiations, and number two, non-ionizing radiations. What are ionizing radiations? So these are all electromagnetic waves which have short wavelength rays. Now they carry enough higher energy to ionize electrons of an atom. The amount of energy present is higher. So that they are normally causing actually the breakdown of chromosomes as well as chromatids. This is called chromosomal mutations. The examples are so high energy to cause breakdown of chromosomes and chromatids. The examples are X-rays, alpha, beta, gamma rays, the cosmic rays. Okay, so these are the different alpha, beta, gamma rays, the cosmic rays, and also the X-rays. That's about ionizing radiations. In comparison with the ionizing radiations, now the non-ionizing radiations have what we call long wavelength. They are long wavelength rays. Now they carry lower energy, unlike what we have just we see that one in the case of ionizing radiations. They carry higher energy. There you have they carry lower energy. Because of the availability of lower energy, they have low or just lower penetrating power than what we have the ionizing radiations. For example, UV rays, the best example for non-ionizing radiations, that is UV rays. Now these UV rays are used to treat unicellular microorganisms, to cause mutations in the case of unicellular microorganisms, or in the case of spores, or in the case of pollen grains. Because in such cases, either in spores or pollen grains, we have the nucleus located near the what is called membrane, near the what is called the plasma membrane or the outer cell membrane. So that's about what we have the radiation, the second physical age. Now we are getting two different varieties of what we have. One is a wheat, another one castor. This is because of what we have radiation. So 2 or 3 lines about the two varieties, what we get because of what we have the mutations. I will write down, and then the final one we have the chemical mutagens. Only the names of what we have the chemicals, how far they are responsible for. Okay. Now the two mutant varieties as given in the book developed because of physical mutagens are number one, Sarbadi sonora. Sarbadi sonora. It is a mutant variety of wheat. It is developed from Mexican variety what is called Sonora 64. And how is it developed? How is the mutation caused? By irradiating gamma rays. Using the gamma rays, now a mutant variety, a beneficial variety, useful variety has been produced. It is a work of, you know that one, M.S. Swaminathan and his team. Now M.S. Swaminathan is aptly called father of Indian Green Revolution. He did so much work for agriculture. That's why he is called father of Indian Green Revolution. Now another mutant variety which is developed by using physical mutagen, that is what's called castor aruna. So it is a mutant variety of castor. How is it developed? It is developed by the treatment of seeds, the castor seeds, with what is called thermal neutrons, a physical mutagen. What for? To induce the early maturity of what we have the plants. To induce the early maturity of plants, that is 120 days instead of 270 days, as seen in the case of normal variety. So these are all the two mutant varieties given in book, which are developed because of physical mutagens. For example, the gamma rays and also thermal neutrons. And that's about what we have the mutant varieties given in the book. Now we have to go for chemical mutagens and co-mutagens. That's about what we have under this category. I want to complete quickly. What are chemical mutagens? Those chemicals that induce mutations are called chemical mutagens. We have a number of chemical mutagens available. Number one, mustard gas, chemically dichloroethyl sulfide. Then we have nitrous oxide, then well known what we have actually the chemical mutagen. Both are efficient in causing mutagens. Then EMS, ethyl methane sulfonate. Then MMS, methyl methane sulfonate, formaldehyde. Then we have manganese salt, used in a kind of dye, and also anthracene, and also ethyl urethane. These are all most of the chemicals. They are used as what we have the chemical mutagens. The effect of nitrous oxide is given in the book. So nitrous oxide normally alters what we have the nitrogen bases of DNA. As a result, they disturb, the nitrous oxide normally disturbs the replication process as well as the transcription process, which leads to the formation of incomplete and defective polypeptides. That is what we have the protein during translation. So incomplete defective polypeptide is formed during translation. This is because of the altering of the nitrogen bases of DNA, which disturb what we have the replication as well as transcription process. That's about what we call the chemical mutagens. And we have what are co-mutagens. The co-mutagens are compounds which are not having actually mutagenic effects. The compounds which are not having what we call mutagenic effects, but can enhance the effects of known mutagens. That's another mutagen. They are called co-mutagens. For example, we have ascorbic acid, which increases the damage caused by hydrogen peroxide. Then another one, what we have caffeine. That caffeine normally just enhances the activity of this methotrexate. So that's about what we have about co-mutagens. And we have some persons also related to the mutagenic agents. I mentioned already H.J. Muller, who is responsible for the introduction of what we have the mutagenic agents for the first time using X-rays in Drosophila. And other scientists also we have L.J. Stadler and also Orbach. And also we have, I mentioned about this dichloroethyl sulfide. It has been used as what we have a chemical mutagen during the World War I. So X-rays was used for the first time by H.J. Muller to induce mutations in Drosophila. Another person, L.J. Stadler, I write down on the board, L.J. Stadler. He was the first person that induced mutations in plants using X-rays and gamma rays. The chemical mutagenesis was first reported by C. Orbach, that is in 1944. So these are some of the persons related to, I write down on the board, you can find out the answer for that one. I already described the co-mutagens. Co-mutagens are compounds which are not having mutagenic properties but enhances the effect of known mutagens. Say an example of that is hydrogen peroxide. This hydrogen peroxide is a known mutagen. Its activity is enhanced by what we have ascorbic acid. So this is a co-mutagen. This is a co-mutagen and this is a known mutagen. So, ascorbic acid, a co-mutagen, increases the damage caused by hydrogen peroxide. This is a original mutagen, but its activity is enhanced or increased by what we have the co-mutagen, that is ascorbic acid. Another co-mutagen, what we have coffin, which increases the toxicity of methotrexate. Another mutagen. Methotrexate is a known mutagen, and this one is a co-mutagen. Coffin increases the toxicity of methotrexate. So this is about what we have the co-mutagens. There are some facts are given in the book. Now the mustard gas, chemically dichloroethyl sulfide. It is used as a chemical weapon or chemical mutagen during World War I to cause mutations in the case of what we have the people of that place. Chemical mutagen during World War I. Then H.G. Muller, I already mentioned about that one. He was the first time used X-rays to induce mutations in the case of fruit fly Drosophila. Then another person, L.J. Stadler, and he reported induced mutations in plants using X-rays and gamma rays. Now the chemical mutagenesis was first reported by a person by name C. Arbach. These are all some of the facts given in the book. So C. Arbach in 1944 just has given the chemical mutagenesis. That is nothing but the production of mutant varieties by using chemicals. That process is called chemical mutagenesis. We already seen what is mutagenesis. So with that I concluded about what we have actually the mutation and mutagenic agents. The first one we have completed the point mutations and mutagenic agents. We have to go for the second type, the chromosomal mutations, in the next class. Okay, thank you. Till then, goodbye.